Thank you, Montse, for your introduction. She said I'm working as a postdoctoral researcher at the University of Évora. What I'm going to talk about today is about myself a little bit, about some of my research fields and my research career. I will introduce briefly some of my previous research, but just briefly, and then I will focus the talk on the research that I've been doing during the last 2 years that consists on the integration of life history traits in climate change risk assessment. Well, as you said, I did my degree in environmental sciences at the University of Granada. Then I moved to the University of Malaga to do my PhD at the Biogeography, Diversity and Conservation research team. This is the group of Raimundo Real. Then I got a postdoctoral fellowship in Iraq, in Ciudad Real. This is a research institute of the CSIC. I was working there with Beatriz Arroyo, and when I finished my fellowship, I continued working there in 3 different projects. And finally, in 2013, I went to the University of Évora to join the Biodiversal project and work there as a postdoctoral researcher as well. So my main research fields are macroecology and biogeography. I think that my previous work can be divided into these 4 sections, like conservation biogeography, applied ecology, climate change, and some methodological papers. They are not, they can be overlaps of a study can be at the same time a climate change study, because we are trying to see the effect of climate change on some species, and at the same time it's a methodological paper, for instance. So just to talk about a little bit about some of them. The main one of conservation biogeography, I think it is the work I did during my PhD. That consisted on the identification of priority conservation areas for vertebrates in Andalusia. So for doing that, I modeled the distribution of all the 325 species in the region, and then applying different conservation metrics and different metrics of physiologic into conservation. We detected the main areas for each of the groups and compared them with the distribution of the natural reserve network of the region. And what we found is that the reserve network was pretty good to preserve this group of species. Another work of conservation biogeography is the identification of priority areas for defragmentations in roads and rail networks. That I did. This was my last work in Ciudad Real. It constituted valuable information to know where we should defragment to put places for the fauna. It was published in a book of the Ministry of the Environment. In Ciudad Real also, I checked some game management activities that are performed in different game states in the region in central Spain, with the intention to increment the numbers of game species, like the red-legged partridge, for instance. So if this game management could have an effect on stepped of conservation concern. And what we got is that none of them have a negative effect, and that some of them have positive effect on the abundance of these species. Related to climate change, we have been detecting the effect of climate change of different vertebrate species, but I'm not going to explain in detail any of these studies, because the majority of them are at the same time methodological papers, because we are always working with species distribution models, try to see how is the best way to forecast the distribution to different areas or different periods of time. So it is like, yeah, I think it's not worth it to go into detail here. But the idea is to try to find the best way of the best selection of variables for modeling the distribution of the species, and how are the best techniques for the extrapolation. One methodological paper that we did was try to check ecological relationships. In this case, we use core types of mammals species in Europe. And we used two different sources of information. On the one hand, we had the distribution in the atlas of European mammals. And on the other hand, we have the IUCN range maps. So both sources of information has problems, has some drawbacks, because in this case, we cannot be sure if absence is really an absence or not. And in this case, what we have is more presences than probably is the real situation, because if there is a gap here, we are going to have just the range map. So what we did was, okay, try to see the core types using this and using that. And what we found is that the relationship was pretty much the same. So the results were concordant, and we think that this constitutes valuable information for biogeographers, because we can be confident that using one or the other will produce more or less the same results, at least for mammals in Europe. And then this is the last paper I'm going to talk about, is the one that we did, where we want to compare the relationship between if the correlation between present-absent models and abundance models were good enough for different species. We did it with two harrier species. So in the first row, we have the maps for the hen harrier, and the second row for the Montagu's harrier. The first column is the models obtained after applying a present-absent model, and the second column is after applying an abundance model, so estimated abundance. So it's normal. It is still under debate if present-absent models can be used to estimate abundance or not. There are many papers about this and with different results. So, and the majority of the times it says that areas of high suitability, at least this, will correspond with areas of high abundance, like here. We are saying that this was the case for the hen harrier, but it was not the situation for the Montagu's harrier, where areas of high abundance were not areas of high suitability. So what we think is that it in this case depends on the level of nest aggregation, because the hen harrier is a territorial species, while the Montagu's is a colonial species that depends on if a breeding pair was breeding in the place before, more than on environmental variables. So with this in mind, and taking into account that I have been performing climate change studies sometimes, but we did not include it. We just included environmental variables, not only climate, also other variables, but just environment. We did not consider any characteristics of the species or any life history trait that will limit the expansion or will help the expansion of the species to other areas. So taking into account these last results and this, keeping this in mind, in this Biodiversal project, one of the main, the project is called European Conservation for the 21st Century. It's very, very general. And one of the main goals was to include these life history traits of the species in microecology and try to see if they can tell us something about the ability of the species to cope with climate change. So there have been some recent papers that have evaluated this. So they have studied what are the life history traits that will help species. At the same time, they detected the more vulnerable species into climate change. And they have been done in different places with different group of species. So the idea is that it is not only that the species will have climatically suitable areas in other places in the future, but that the species may have the ability to reach this area, to span until this area. So how can we include that? So not only climate, but also non-climatic limitations. So we wanted to test this in plants, mammals, and birds at the European level. Sorry. And normally, dispersal ability of the species is the main trait. because the species may have the ability to disperse to this new area. So in some studies, this is a review, dispersal is included, but sometimes it's very difficult to include a real value of dispersal data for species. So what we normally do is say, okay, this is going to be climatically suitable areas in the future, considering that the species will reach everywhere, so that full dispersal in this case. Or we are going to consider that the species is not going to move, so no dispersal at all, or a partial scenario of dispersal. But these are like, I think that this improves just using just climate change and that's all, but it's not enough. And apart from that, we consider that we were interested to include not only dispersal, but other traits that were related to in the range shift process. So it is that the species may have the dispersal to arrive to a new place, then to have the capacity to establish in that place, and finally to proliferate after the establishment. And then we have more dispersers to go to other places. So this was the idea, try to find traits related to these three elements of the range shift process. So our database was consisted of 1,275 plant species. We just work with native species, 125 mammals, and 335 birds. I'm going to show you the methodology of all of the studies all together, but just to let you know that I did one separate model for each of the groups, not combining all the groups together, one for plants, one for birds, and one for mammals. So first what we did was a group meeting to try to see, to try to decide where are the important traits. So checking in many different places in the literature and talking about with some people, we decided like we wanted to check like almost 20, to find almost 20 traits for each of the group. So there are many places where we can find this information. Many of them are available on the internet. Others you have to do like an agreement with the owner of the data, but it's more or less easy. But then we also check on books, on different papers, and in total for these three groups we checked more than 150 sources to find the traits. It's just to let you know that this is very hard work and time consuming, but fortunately we have a technician in the project who did the majority of this search. So we aim to find all the information, we found that in the case of plants, although we were interested in 20 traits at the beginning, we only have enough information for 12 of them. Here you can see the 12 traits that we use for plants, and in parentheses you see the number of species. The main drawback when you are working with traits at this level is that the number of species is not the same for all of them. So it's very difficult to, okay, if you say, okay, I'm going to see how many species I have with information for all the traits. So if you remember, I have more than 1,000 species, but when I did that, it was like 5. So it's impossible to do a model with all the species together. So it just for you to keep in mind when I explain the modeling technique that it is, you have to do it step by step. It's not possible to do a multivariate model since the beginning. In these traits, I'm not going to read all of them. You can do it if you like, but we have traits that are just a value, a continuous variable, like specifically varia or plant height. We use just one value for each species. So in this case, it's the mean value, in other is the maximum. It depends on the trait, but it's one value for species. Then we have others that we have to calculate it, like this habitat breadth is an index that we perform considering the name of habitats where the species can live in. So we did an index combining the general habitats and the subhabitats. When I mean general habitat is like forest or grassland, and subhabitat is the type of forest, for instance. So considering all of them, we make an index. Then we have dispersal distance. This is the most tricky one. because we did it with following this paper that you cannot see, but it's the one of Vitos and Engler in 2007. And they proposed a way to classify, to establish a category of dispersal distance for each plant species according to many different things. For instance, according to the vector that can disperse the seeds, according to the number of seeds the plant can produce, according to the height of the plant, all the type of things. So we need to have all this specific information and combine them according to what they say, and obtain finally a value of dispersal distance that is an ordinal variable with seven possible classes. It's not a real value either of dispersal distance. Then we have some categorical variable, that's seed bank persistence, that it consists of 3 possible values. This trait is related to the how much time the leaves, the seeds, sorry, can be viable in the soil. So we have 3 possible values: less than 1 year, between 1 and 5, or more than 5. So just to let you know that the variety of things that we have here. And in the case of birds and mammals, again, we were interested to have traits of dispersal, establishment, and proliferation. And the original idea was to have common traits for both groups, although we are analyzing them differently, but separately. But what we have is that we have these common traits for both of the groups, but for some traits we don't have enough number of species for any of them, so we include it in one group, not in the other, always due to the data, for the problem of data availability. So here we have like traits related to ecological generalization, habitat breadth or diet breadth, with the body size, brain mass, and with the speed of life history, like sexual maturity, age, fecundity, etc. Another problem that we have here is that we don't have real dispersal data for mammals or birds, because there is not enough information. So we only could check it indirectly about migration. That's the only thing that we can do. So we have all the traits. Okay, one thing I want to say is that in the case of animal groups, we for some traits we check for the we do an allometric correction of some traits, because bigger mammals, for instance, will have bigger brains. Is normal. So we to estimate, to have a value of the trait of brain size, what we did was a log linear model between brain size and body mass, and the residuals of that model were the values that we use for this trait. So this was about traits, and now about ranges. We have the data of the ALARM project, that is a European project that was finished. In this project, we have the distribution of these species at 50 by 50 kilometers squared in more or less Europe. It's not called Europe, but more or less. And the climatic variables are the same resolution. So we use the geographical ranges as response variables, and we use 2 possible ones. One was the range size, just the number of the cells where the species is living, and range filling. That is the proportion of climatically suitable areas that the species can occupy. So to calculate range filling, we need to calculate the potential range. So what is the potential distribution for the species? You can have it with a species distribution model, for instance, but in this case we did a PCA environmental approach. So here is a schematic representation. You have we did a PCA with climatic variables, and the 2 axes of the PCAs are here. So this is the environmental space. In orange, we have the environmental space that is available in Europe, and in different colors are the different species. For instance, for the blue species, we have that the dots are the presences, and the polygon is the potential range. And the range filling is just the ratio between presence and potential. That's so we did it with the 100% of the species occurrence, and just also with the 95%, because we wanted to avoid some biases related to a species living in very extreme climatic conditions for any reason. And what we think is that this range filling can tell us something about the range ability of the species. Because if it were just by climate, the species potentially could reach all the potential range. But if it does not happen, because some species have higher range filling than others, this is because there are other factors that are limiting this expansion. We consider that some of these factors could be the life history traits. So this is the reasoning that we are using the range filling. So we think that species with higher range filling are those that are more able or have been more able to expand their ranges will be the one that will be more able to cope with climate change. This is the idea. So what we did was use range size or range filling. This is not a ratio, it's one or the other, as responses and life history traits as predictors. So as I told you at the beginning, we need to do this in different steps. First of all, we perform univariate models and selected the significant, those that have a significant relationship with the response variables to include it in the next multivariate models that we did also in 2 steps. I'm not going to go into detail, but yeah, finally you need to select the subset of traits in different steps due to data availability. So in the case of plants, what we finally did was an average model according to the archaic information criterion, selecting the subset of models with a difference of AIC lower than 2 units. And what we found is that range size and range filling, we have also range filling 95 here, but I'm not going to show you because it's almost the same. So range size and range filling, the traits were the traits that were important for range size were also important for range filling. We don't have any difference. And the ones that have the highest relative variable importance of 1, that this is what's included in all the boundaries of the subset, were dispersal, seed bank, and habitat breadth. I prefer to show you this in a plot rather than in a table. This is the same table as before, because for me, it's not, I don't know, it's like more friendly to explain it here. But I want you to know that these are not univariate relationships, but the relationships that are in this table. So in this graph, you always have to think that on the y-axis is the range, can be range size or range filling, and on the x-axis is the trait. We have just one plot because the same traits were important for range size and for range filling. So I'm just showing one scenario. So in the case of plants, those that have higher dispersal ability, that can live in a variety of habitat types, and that can have seed banks viable in the soil for more years, are more able to cope with climate change. So in the case of dispersal, this is like a expected result, but at least we could check that our metrics of this ordinal variable was also reflecting the capacity of the species to disperse. Then the seed bank, we have that this result that species with longer that can maintain seed bank for more time, for more years, are well positioned to cope with climate change. This is also a logical result because they can be persistent in unfavorable conditions for some years. This was a pretty new result because this was analyzed before in more local studies, but it was one of the first time that the persistence of the seed bank were detected as important in a macroecological study. And then in the case of habitat breadth, we were expecting a linear positive relationship, but we have these unimodal results, either here or in univariate models. We found that some authors detected that some generalist plant species tend to be competitors, while specialists tend to be stress tolerant in this CSR spectrum, competitor stress tolerant ruderal spectrum. So what we consider is that probably this unimodal response in our case may be also related to a trade-off between tolerance to stress and competitive ability. So these were the main four traits for plants, but probably you are thinking that species are not independent. So we took also into account the phylogeny of the group, and we test phylogenetic autocorrelation. And here we have in black dots the observed variables, range size of range filling, and in white the residuals of the model. So we can see that the observed autocorrelation, for instance, in range filling, is not maintained when the model is applied. Moran's I value are near zero, nearly zero. It is not significant, so we can be happy that we don't have problem of the phylogeny in our results. And then we also check the difference between the GLMs and the PGLS. So a model considering the phylogeny with P or not considering the phylogeny, and we obtained that there were no difference in log likelihoods or in the significance of the variable. So well, we are happy that we don't have problems with the phylogeny in our results. So this is for plants. And for animals, we have this result is that, well, this is also numbers in a table, but I'm just showing you these plots. In the case of mammals, it is the only group that we found a different relationship of traits for range size and for range filling. In the case of birds, we have, again, the same pattern for the two response variables. Range size, mammals that have higher ranges are those that are more generalist, both in diet and in habitat. Then habitat breadth was included in both groups of range filling. That is, ecological generalization is also important for animals to cope with climate change. It's normal because they can live in more different places. And then we obtained that traits related to the speed of life history, like species early reproducing or species with higher fecundity, are the ones that will be we will have less problems in an event of climate change. In all these cases, in mammals, birds, and in plants, the deviance plans is higher than 30%. It's not too much, but not too low. And well, I think that's all. Yeah, so we can conclude that we have obtained the same consistent result with the univariate and the multivariate models. I didn't show you the univariate results, but the relationship were pretty similar with the ones obtained in the multivariate models, and in this case we could have like 600 species and here just 100, so it was not expected, but yeah, we obtained strong relationships. And then we think that the specific values that the specific traits that we have obtained as important can be included in climate change risk assessment, and that in climate change risk assessment not only dispersal but also traits related to establishment and proliferation should be considered, because we have shown that they are important as well. So I've been reading a lot about this. So we are doing an opinion review paper with all these things in mind, trying to see where we can find traits that are telling us something about the ability to cope with climate change. I mean, I show you range size or range filling, but we can use also range shift or population trends of bird species, for instance. So you can combine this with traits, and you could have important traits. So try to find where we can find all of them. Then we are proposing a framework to group the traits, because we acknowledge that it is very important to see if a trait is important for the establishment or for the proliferation process. It's like almost impossible to distinguish. So we think that with these 7 groups, dispersability, ecological generalization, avoidance of small population effects, persisting in unfavorable conditions, speed of life history, competitive ability and site fidelity, will be more than enough, at least for this group, to put traits in some of these categories. And we consider that it's more important to use different traits belonging to different groups that for traits belonging to the speed of life history, because probably it's going to tell you the same. And our next step is to do to establish a threat classification of the species based on the risk of that the species have to climate change and fragmentation, and to the vulnerability to this risk. So we can imagine, we can try to put this in this diagram. So here we have how is the climate change and habitat change in the place where the species is living today. Here is climate change in future area, or habitat, suitable habitat in a different area. And then we can plot in the bottom-up axis the ability of the species. So this bottom-up are the traits. So this will be a species that have traits, if it is here, that has the ability to change, and here to be maintained in the same place with different climatic conditions, for instance. So we can establish like different risk syndromes for different species. So and we can plot all the situations for all of them. So in the first case, for instance, here we have that climate change is not affecting the species too much, fragmentation either, and the species has the ability to shift the range or to be maintained in the same place. So everybody would like to be like this one, because it has no problems at all. But then we could have this scenario where we have like an important effect of climate change on fragmentation on the species, and the species have not the ability to shift the ranges because it has not the traits that we have considered important. So we could plot all the different combinations, and finally, we think that we could establish a category of risk for each of the species that could be used in conservation as well. And I think that's all. I want to thank you, my collaborators. I want to thank you, Montse, and all the members of the groups, because I'm feeling here like at home since the first day. Thank you for your attention.